Hello everyone. So once again welcome back to my channel. So in this session I'm going to discuss about post fertilization changes in angiosperms. So first to start with, we will discuss a table showing post fertilization changes, a table which gives the idea about post fertilization changes. So first, before fertilization and after fertilization, what are the changes taking place? So first, calyx and corolla, they wither away, ovary will change into fruit, ovule into seed. Integuments, outer integument will change into outer seed coat, which is called as testa, and inner integument will change into inner seed coat, that is called as a tegument. Micropyle will change into seed core and funicle will form the stalk of the seed. Nucellus, normally it will be consumed, but in case if it is present, it is called as a perisperm. In case if there is some remnant of nucellus present, then it is called as a perisperm. And egg cell which is present in the embryo sac, that will change into zygote, and synergids will degenerate. Hilum will form the scar of the seed. Now we can also give the ploidy of each part here. Suppose we take the ploidy of fruit, say ovary changing into fruit, we can give the ploidy will be 2n. Similarly here also 2n seed coat, and all these tegument will be 2n. Seed pore, which is just an empty space, we can give 0 ploidy. Again stalk of the seed will be 2n. Perisperm also 2n since it is formed from the nucellus. Zygote will be 2n, and synergids, since they degenerate, synergids all will be n. And hilum, which belongs to the ovary, scar of the seed, this also will be 2n. So this table will give you the complete how the transformation occurs between the ovule and the seed and post fertilization changes. Now all these events which are taking place after fertilization, that we call it as a post fertilization change. Now the most important and the first event which is taking place after fertilization is formation of endosperm. Endosperm formation. This is the first post fertilization change which is required. It is preceded by, it is preceded by embryo formation or embryogenesis. It is preceded by embryogenesis. Preceded means it takes before the formation of embryo, because for growing embryo nutrition is required. So embryo will be formed first, then followed by endosperm will be formed first, then followed by embryo. So endosperm formation takes place first. So in angiosperms, if we observe, the endosperm is formed from primary endosperm nucleus. This primary endosperm nucleus is the result of triple fusion. The primary endosperm nucleus, it is the result of triple fusion. Triple fusion is fusion of male gamete with secondary nucleus which is present in the embryo sac. So primary endosperm nucleus is formed. This is present in primary endosperm cell, which is the central cell. So primary endosperm cell, it is the first cell of endosperm. It is the first cell of endosperm, and it divides to give rise to endosperm. And in some organisms, in some members of angiosperms, like orchidaceae members, orchidaceae members, or podostemonaceae members, here the endosperm is not formed at all. These are monocots. The endosperm is not formed at all. Triple fusion will occur and primary endosperm nucleus is formed. Further, Primary endosperm nucleus will not divide and it will not form any endosperm. So they can be called as non-endospermic seeds. Normally, endosperm means angiosperm is a triploid because it is formed as a result of triple fusion. Three haploid nuclei are fusing. So it is formed as a result of triple fusion. But in gymnosperms, the endosperm is haploid. Endosperm is haploid because it is directly formed from female gametophyte without any fertilization. It is directly formed from female gametophyte. So that's why we can consider here endosperm is haploid. Now, next we will see about what are the different types of endosperm based on their formation. Okay, now let us now continue with the what are the types of endosperms. There are mainly three types of endosperms: nuclear endosperms, cellular endosperms, and halobial endosperm. So we will see what is the difference between these three in terms of their formation. So nuclear endosperm, like we find in case of maize, wheat, rice, sunflower and capsella. So here what happens is, first the primary endosperm cell. This is the primary endosperm cell which initially undergoes free nuclear divisions. It undergoes free nuclear divisions. Free nuclear divisions means only mitotic division without cell wall formation. So it undergoes only free nuclear divisions. So these free nuclear divisions, all the nuclei will be present. It becomes a multinucleated state, After all nuclear divisions are complete, then cell wall formation occurs. So when cell wall formation occurs, cell wall is formed in centripetal direction. That is, it starts from periphery and proceeds towards the center. So we find all these nuclei, they are covered by the cell wall. So cell wall formation starts from periphery and it is moving towards the center. Finally, endosperm becomes completely cellular. There is one exception in case of coconut. In case of coconut, we have coconut water and coconut meat also. The white kernel which is edible, and of course even water also. These belong to the endosperm. So coconut water and coconut meat. So here both the types are there. So here what happens? The coconut water is due to free nuclear divisions. Only free nuclear divisions will occur without any cell wall formation. Whereas the coconut meat or the white kernel coconut meat, this is due to cell wall formation. That means cell wall formation starts centripetally and it is not complete. When it is not complete, coconut water remains at the center, that is in the form of free nuclear division. Wherever there is a wall formation, we find a solid part is formed, that is coconut meat, or it is also called as a kernel. So this is example of both nuclear as well as cellular endosperm. Now the second type we have is cellular endosperm. In cellular endosperm, the cell wall formation takes place right from the beginning. So suppose first a nucleus divides and forms two, immediately it is followed by cell wall formation. So cellularization, what is cell wall formation? Also called cellularization. So here we can say cellularization Cellularization takes place from the beginning. Takes place from the beginning. So that is called as a cellular endosperm. Like for example, it is found in Datura, Petunia, and Petunia. So here it is cellular endosperm. Now, next type, the third type is called as halobial endosperm. Halobial. Halobial is a combination of both. It is a combination of nuclear as well as cellular. So here what happens is, initially the primary endosperm cell will divide to form two unequal cells. Two unequal cells are formed. One is chalazal, this side it is called as chalazal, and the other one is called as a micropylar. Chalazal and micropylar. So now micropylar will further divide and form the endosperm. That means it undergoes cellularization. Whereas chalazal will undergo free nuclear. It may or may not divide, and if it divides, it can undergo free nuclear. So it is a combination of both nuclear as well as cellular. So we will just go through once again what are the examples. Now let us look at the examples of the endosperm, that is halobial endosperm. So halobial endosperm, it is found, example is asphodylus. It is found in some monocots. It is the feature of some monocots. Halobial endosperm, mainly belonging to the order Halobiales. That's why it is called halobial endosperm, belonging to the order Halobiales. So these are the three types of endosperm. Now we will continue with what is the fate of endosperm. Fate of endosperm. That means what happens to endosperm after it is formed. So it is consumed by the developing embryo. We know that the endosperm is formed for embryo. So for consumption for embryo, it is formed. Embryo must be consumed. Now based on that, there are two types of seeds formed. One is called as a endospermic seed. Endospermic seed, or this is also called as albuminous seed. Endospermic seed, or this is also called albuminous seed. So here a part of endosperm will be left. It will be consumed and some part of endosperm will be left. Like this is generally found, example, in monocots. It is generally found in monocots. So any monocot, you can give the example of endospermic seed, like cereals, any example of cereals. But there are some dicots also which show endospermic seed, like the members of Solanaceae. These are the exceptions. Members of Solanaceae and also castor. These are the dicots having endospermic seed. Normally endospermic seed is found in monocots, but rarely in some members of Solanaceae and in castor, endospermic seeds are present, though these are dicots. Now, the second type are called as non-endospermic seeds. Non-endospermic seeds. So here endosperm is completely consumed by the growing embryo. The embryo will completely absorb the nutrition and almost no endosperm is left. So this is also called ex-albuminous seeds. These are also called ex-albuminous. Normally dicots. Dicots are normally, they do not have endosperm. Like members of, you can take any members of Fabaceae, like bean or pea or any member of Fabaceae. These are the non-endospermic seeds. Now, sometimes the nucellus will be left behind. Normally, nucellus is consumed by growing embryo. Nucellus, gives food to the embryo sac. So, normally, just before fertilization itself, the nucellus will be consumed. But in some, the nucellus is left. That is called as a perisperm. So, perisperm is remnant of nucellus. Perisperm means it is a remnant of nucellus. So therefore, it is diploid. This is therefore diploid in nature. Some perispermic seeds, like for example, you can take example Piper nigrum. Piper nigrum is pepper, black pepper. That is Piper nigrum. And another example, you can take Nymphaceae like Nymphia or Nilobium, that is lotus. And another example, you can take that of the coffee, beetroot, coffee. So all these, they also contain perisperm. The food material is stored in the perisperm. So perisperm, some like in Piper nigrum we find both perisperm as well as endosperm. Both are present. So with this we conclude up to the endosperm, types of endosperms and perisperm. In my next video I am going to discuss about the development of dicot and monocot embryo. So till then, have a good day.